Hello everyone. My name is Nausheen Parveen, Assistant Professor in IIMT College of Pharmacy, Greater Noida. In today's lecture, I'm going to discuss about chemotherapy of malignancy. First, cancer or neoplasm. Cancer can be defined as a mass of tissue formed as a result of abnormal, excessive, uncontrolled, autonomous, purposeless proliferation of the cell. Cancer is a disease characterized by abnormal, uncontrolled cell division attacking the surrounding tissue and organ and also the distant body part by circulating with blood and lymph. Neoplasm or tumor is an abnormal mass of tissue resulting either from uncontrolled division or from obstruction of cells. What is the neoplasm? Neoplasm may be two types. First is benign tumor. Second is malignant tumor. First is benign tumor. Benign tumor is it is an non-cancerous cellular growth which does not invade the nearby tissue or spread to other body. Malignant tumor is a cancerous, undefined, unpredictable cellular growth which invade the nearby tissue directly by the lymphatic system or circulatory system. Next is introduction. Cancer is a group of disease involving uncontrolled cell growth. A normal cell turn into the cancer cell because of one or more mutation in its DNA which can be acquired or inherited. Cancer arises as a result of a series of genetic and epigenetic changes, the main genetic lesion being inactivation of tumor suppressor gene, the activation of oncogene. Next is characteristic of cancer. Cell characteristics that distinguish them from normal cell: uncontrolled proliferation, deep differentiation and loss of function, effectiveness, or next is metastasis. Next is the etiology. Etiology, environmental toxin, smoking, tobacco, azo dyes, benzene, asbestos, papilloma virus, STLV1, UV rays, phthalates, or perfume. These are the etiologies. Next is the type of cancer. Cancer is a anal cancer. Affected area is anus. Breast cancer is affected area is breast. Bladder cancer affected area urinary bladder. Bone marrow cancer affected area shaft of long bone. Colon cancer affected area colon. Cervical cancer affected area cervix. Eye cancer affected area is eye. Gynecological cancer, female reproductive cancer, female reproductive organ is the affected area. Next is lung cancer, affected area is lung. Osteosarcoma, affected area is metaphyseal region of long bone. Next is Wilm tumor, affected area is kidney. Leukemia is the next type, affected area blood. Larynx cancer, larynx, affected area. Next is testicular cancer, affected area is testes. Rectal cancer, affected area is rectum. Next, it is a growth phase of cancer cell division. It is a cell cycle. First is G1 phase, which is related to cellular contents, excluding the chromosomes are duplicating. Next is the S phase, which is related to each of the 46 chromosome is duplicated by the cell. Next is G phase. The cell double check the duplicated chromosome for error, making any needed repair. Next is the mitosis. Next, pathophysiology of carcinogenesis. First is a normal cell convert in DNA damage by by failure of DNA repair. Next is the next step, mutation in genomes of somatic cells. And next, activation of growth promoting oncogenes, alteration of gene that regulate of apoptosis, and inactivation of cancer suppressor genes. Next, expression of altered gene product, loss of regulatory gene products, and then cause malignant neoplasm. Next, the cell component of the solid tumor. First component is component A, B, and C. First component A consists of dividing cells, possibly being continuously in the in cell cycle. Component B consists of resulting cell Z0 phase, which although not dividing are potentially able to do so. Component C consists of cell that are no longer able to divide but which contribute to the tumor volume. Next, only cell in component A which may forms a little as 5% of some solid tumor are susceptible to the main current cytotoxic drug, which affect only only one characteristic aspects of cancer cell biology is called cell division. Next, in many cases, the anti-proliferative action is during S phase of the cell cycle, and the resultant damage of the DNA initiated apoptosis. Because most of the anti-cancer drugs are anti-proliferatives, they affect rapidly dividing normal cells and are thus likely to depress growth. Most causes nausea, vomiting, hair loss, and teratogenicity. Next, cancer therapeutic model. There are three models: surgical, radiation, and chemotherapy. So in surgical, one third of the patient without metastasis respond to surgery and radiation. In radiation, if diagnosed at early stage, close to 50% cancer could be cured. And chemotherapy, 50% patient will undergo chemotherapy to remove micrometastasis. However, chemotherapy can be cured only about 10 to 15% of all cancer patients. New type of cancer treatment. First is hormonal treatment. Next, specific inhibitors. Next, antibodies. Next, biological response modifier, and vaccine. First one is hormonal treatments. These drugs are designed to prevent cancer cell growth by preventing the cell from receiving signals necessary for their continued growth and division. Example, breast cancer, tamoxifen after surgery and radiation. Next is specific inhibitor, drugs targeting a specific protein and process that are limited primarily to cancer cell or that are much more prevalent in cancer cell. Next is antibodies. The antibodies used in the treatment of cancer have been manufactured for use as drug. Example, Avastin. Next है biological response modifiers. The use of naturally occurring normal protein to stimulate the body's own deficiency against cancer. Example है rituximab. Next, vaccine stimulates the body's deficiency against cancer. Vaccine usually contain proteins found on or produced by cancer cell. By administering these proteins, the treatment aim to increase the response of the body against the cancer cell. Next, malignancy, which responds favorably to chemotherapy. First is choriocarcinoma, acute leukemia. Third is Hodgkin disease, Wilms tumor, testicular carcinoma, retinoblastoma in children, and last had diffuse histocytic lymphoma. Antineoplastic drugs are the most effective against rapidly dividing tumor cells. General toxicity, nausea, vomiting, bone marrow depression, diarrhea, immunosuppressants, alopecia, delayed wound healing, carcinogenicity, hyperuricemia, testogenicity, neuropathy, cardiomyopathy. Next है classification of anti-cancer drug. First is the cell cycle specific and second one is the cell cycle non-specific. First, cell cycle specific drugs act on proliferating cells, are most effective in the hematological malignancy and in solid tumor in which a relatively large proportion of the cell are proliferating. Next is cell cycle non-specific drugs can be kill both G0 and cycling cells, although cycling cells are the more sensitive, are used in showing slow growing tumor. Cell cycle cell cycle specifically of anti-neoplastics. These all are the drugs which is act on specific phase, like cell cycle phase, like G1 phase, actinomycin D, 5-fluorosil, cytosine, arabinocytes, methotrexate, 6-mercaptopurine, 6-thioguanine. All drugs are at on G phase. And next is the S phase, purine antagonist, methotrexate, cyclophosphamide, 5-fluorouracil, cytosine, arabinocytes. These all drugs are act on S phase. And next is cyclophosphamide, bleomycin, actinomycin D. These drugs act on G2 phase. Next is vincristine, vinblastine, paclitaxels. These all drugs act on M phase. Next, classification of anti-cancer drug. First class is cytotoxic drug, and subclass of cytotoxic drugs is alkaline agent. Example, maclothiamine. Next is nitrogen mustard. Examples are cyclophosphamide, isofosamides, melphaline. Next is ethylamine, thiotepa, alkyl sulfonates, busulfane, nitrosourea. Examples are carmustine, lomustine. Next is the triazine, decarbazine. Next is methyl hydrazine. Example, procarbazine. Next class of drug of cytotoxic is platinum coordination complex. Examples are cisplatin, carboplatin, oxaplatin. Next class, antimetabolites. Subclass, first one is folate antagonist, methotrexate. Next is purine antagonist, 6-mercaptopurine, 6-thioguanine. Pyrimidine is the next class, 5-fluorouracils. And next subclass of cytotoxic drug is microtubule damaging agent. Examples, vincristine, vinblastine. Next is the topoisomerase 2 inhibitor, etoposides. Next, topoisomerase 1 inhibitors, topotecan. Next है antibiotics. Examples: actinomycin D, mitomycin C, doxorubicin. Next is miscellaneous: hydroxyurea. Next class, targeted drug, tyrosine protein kinase inhibitor, targeted drug, tyrosine protein kinase inhibitor, imatinib, EGF receptor inhibitors, irlotinib, angiogenesis inhibitor, sunitinib, proteasomes inhibitors, bortezomib, unarmed monoclonal antibodies, rituximab. Next next class is hormonal drug. First is glucocorticoids, prednisolones and others. Next estrogen. Examples: ethyl estradiol. Next class, selective estrogen receptor modulator, tamoxifen. Example: next is selective estrogen receptor down regulator. Example: fulvestrant. Next, aromatase inhibitor. Example: एक्सीमेटेन. नेक्स्ट है एंटी एंड्रोजन, फ्लूटामाइड. नेक्स्ट 5 अल्फा रिडक्टेज इनहिबिटर, डस्टास्ट्राइड. GnRH एनालॉगस. एग्जाम्पल: ल्यूप्रोलीन. Next class, progesterone. Example: hydroxyprogesterone acetate. First class, alkylating agent. Attach alkyl group to DNA, allow cross-linking of base pair, damaging DNA cell cycle, non-specific. Typical alkylating agents: cyclophosphamide, isopharmides, melphenol, busulfane, macloridine. These are the examples. Side effects: nausea, vomiting, myelosuppression, secondary malignancy, infertility, hemorrhagic cystitis. Next is the typical alkylating agent: platinum compound, covalently bind purine DNA basis drugs, cisplatin, carboplatin, oxaliplatin. These are the drugs. Next, mechanism of action of alkylating agent. Alkylating agent form highly reactive carbonium ions and transfer alkyl group to nucleophilic sites on DNA basis and result in the cross linkage, abnormal base pair, DNA strand breakage, and decreased cell proliferation. Next है mechanism of action. Platinum coordination compound that inhibit DNA synthesis, cross-link and denatures stand of DNA, disturb DNA function, and covalently binding to DNA bases can also produce DNA intrastrand cross-linking and breakage. Mechanism of action of cisplatin. Cisplatin enters cells, form a highly reactive platinum compound, and intra-stand and interstand cross linkage then cause DNA damage and inhibit cell proliferation. Antimetabolites. Next, antimetabolites inhibit DNA replication or repair by mimicking normal cell compound. Next is folate inhibitors. Methotrexate is the example for prevent regeneration of THF, adjuvant leukotriene to protect healthy cells. Side effect is mucositis. Next is the pyrimidine inhibitors. 5-fluorouracil is the example. Bolus does cause myelosuppressor. Continuous dose causes GI problem. Next is the capacity side effect is hand-foot syndrome. Palm and hand and feet become red and can start blistering. Next is the microtubulin targeting agent. These drugs inhibit mitosis, specifically M-phase. Vinca alkaloid destroy microtubulin, obviously preventing their function. Vincristine and vinblastine and vinorelbine. Side effect is peripheral neuropathy. Next is the taxane, preventing their functions. Doxetane is the example of taxane. Side effect is myelosuppression, peripheral neuropathies, hypersensitivity from diluent, camphor dilute in paclitaxel. Avoid hypersensitivity with arabaxane. Next is the topoisomerase inhibitor. Topoisomerase 1 inhibitor prevent relaxation of supercoiled DNA. Example, topotecan. Both have side effect of myelosuppressor. Next is the topoisomerase 2 inhibitor prevent recoiling of the DNA after transcription. Etopocytes both have side effect of myelosuppression. Anthracycline. Various mechanisms of action inhibit topo second generate ROS, perhaps alkylation. Next है monoclonal antibodies. Origin determined from suffix OMAG from mouse, and CMAG is chimeric. Rituximab targeting site CD20. Treatment lymphoma. And these are the other chemotherapeutic agents.